Good morning, good evening, g'day. I hope you are well where you are listening. This is a medical cannabis program. I'll be talking about legal practices as I respect the law and hope you respect the law too. And talking about the safe uses and consumption and growing and breeding and all that sort of stuff to do with medical cannabis. Woohoo. Good meds for everybody. And today was an open topic. There it is, open topic today. So I was waiting for some comments and hopefully there will be some suggestions on where to go or what if somebody needs some help in some ways. I can show some slides to familiarize with the topic and go from there. Otherwise, I'll be showing you stuff that I want to look at that you mightn't be as interested in. But I was thinking about going on down the lines about what a plant knows, how it hears, how it feels, all that sort of stuff, and discuss that. That's what I was thinking of. Going down that route if there's no suggestions. So I'll just wait a little bit. If there's no suggestions, I'll start doing that. It's pretty interesting what a plant, it's amazing on how you just think, oh, you're just looking there, it's just sitting there. But it actually can do a lot of things and can interact with the environment. It's really fascinating. It was a pretty cool, it's from a course I'll be going through. It's a course that was done from Tel Aviv University, and I'll be showing some slides from that. And it was quite informative actually. It was a good course. All right, well, I'll get into it. Supreme Grape, hello. G'day. I'm going to start talking about how the plant can hear and feel and touch and sense and all that sort of stuff. I was thinking, unless there's some suggestions here, I'll chat slowly starting to come in. Good morning, Ned Kelly, how are you? Bamboo One, hello Bamboo One. Bamboo One says, hey AussyCC, how things mate? Oh, yeah. What's your opinion on electroculture gardening? Electroculture, that's to do with electricity, I reckon. Electroculture. Never heard of it, to be honest, but electricity, maybe through vibrations. Electroculture, is it that technology? Because I've heard about that one, but I haven't specifically heard about electroculture, mate. Sorry. Can you elaborate a little bit? Hello, Jimu Sema. Hey, no suggestions. No suggestions. That's okay. Well, I can talk about what a plant knows and how it feels, and go through that course. Unless because in Bamboo's question, it touches on that, on what a plant feels. Did you know it responds to vibrations? Plants do. They grow towards it. They put out a calcium signal that makes them grow towards the signal, and it's the frequency is in the base hertz. It's 200 hertz frequency. So if you play a lot of your stereo with a lot of bass music, it can vibrate and the roots can get attracted and they grow towards it through that calcium signaling. That's a technology that's been patented in the States through vibrations, and it also helps vibrations with anyway, I should just rip into the slides instead of just looking at a blank screen, eh? Yes. All right, hopefully that question, Bamboo, will be explained if it's to do. There's a few things related to that vibration technology in these slides. Share screen. What's this one? So that's all these slides. We should go big, I reckon. No, don't need to know receptors. Actually, the thing that says the amounts. Yeah, this one. So the difference in the receptors for sight for plants can see, because that's how they grow. They're autotrophs. Medical cannabis is an autotroph, and it responds primarily in the 750 nanometers to 300 nanometer spectrum, from the far red to the infrared, down to the UV spectrum. And humans are just above that. We respond a bit better. We see in our light from 400 to 700. This is the difference here between plants and humans in their sight. So plants they're sensed in all of the cells, where ours is only in our eyes and our retina. And then the plants has 12 photoreceptors, which we can go back here and I'll go through them briefly in a sec. I'm not going to touch on because that's a bit technical. And humans have 5. In plants, for sight, it enables finding energy for food. That's how they use it, because they're an autotroph, so they need to sense that the right spectrums are coming in for them to photosynthesize and metabolize the energy out of it. And humans, it identifies details, the sight. For in plants, it synchronizes the biological clock and seasons of the year, like circadian rhythm. Plants respond very well to the circadian rhythm, and same with humans. And back here to what the receptors are. So these are the different receptors. So it's primarily in the blue and in the red spectrum, and they're called cryptochromes in the blues and phytochromes in the reds. And then you can break down, then you go into an UVA, there's different UV receptors too. I should have said as well, but that's near the top of the blue. That'll do. The different tropisms, actually, primarily the photoreceptors for tropisms, they're not really shade. To get shade, it picks up on shade, the phytochrome, so it picks up on the red, because what it does is the light comes through and it'll go through one leaf, and then it'll use most of it, and the bit that comes out through it is the red. So it senses the phytochrome, which is the red receptor. That's how the plant gets it through shade. That's how it puts it through to sleep as well. So you can get different, there's an experiment. I wonder if this touches on those. Actually, there's quite a few slides here to go into, so I'm not going to elaborate here. So these are the primary photoreceptors. So transition to flowering, because they've got to use that to know that they use their photoreceptors for the light to drop down from a 18-hour period to about a 12-hour period. To germinate, some need a little bit of light, because they'll grow and develop chloroplasts. And if they don't, they end up being all white, and that's eleoplast, when it grows and there's no pigment yet, because it hasn't been able to photosynthesize from the sun. That's the next one too. Good. The difference between the humans and plant cells is not much difference, believe it or not. They're pretty similar. It's only 3 differences. And in plants, they've got the chloroplast to develop that's the green pigment for photosynthesis. They got a vacuole, stores stuff, and they got a thicker cell, an extra cell wall. Ethylene influence. So this is an ethylene influence. Ethylene is one of the 5 basic plant hormones, and this is its influence if it's grown in dark. It'll grow, the more ethylene, the more shortened hypocotyl, and it's called a triple response, where it's a shortened hypocotyl, it's thicker, and it has a hook function, a hook process. That's how you can know when you're breaking dormancy. You'll add ethylene, because it's one of the signaling pathways of the germination process, as well as gibberellic acid. But you'll add ethylene through an apple or banana just sitting there, which emits it. And if you've got too much, this is the effect. You'll get the triple response. What's this one? Without ethylene. That's a bit technical. It's technical as. This is how the plant speaks to another plant. It forms a stress hormone, salicylic acid, and then it makes it turns it into a methyl salicylic acid, which is the volatile form, and then that's how it goes into the air. And then another plant's receptors pick it up, and they pick up this methyl salicylic acid and exchange it back to salicylic, and then activate its pathways through other plants. So that's how one plant speaks to the next plant, and it's called allelopathy. Oh, I see what's going on. Look at the screen there. I want it to be this screen. How do I show just the screen? I just saw that. I'm looking on the tablet. G'day, Dave. Oh, there's a few other suggestions. It's when they put a copper-like antenna in the ground, drawing energy from the air. Holy mackerel. Electroculture. What could that be happening? I have an electrical engineering background as well. So they're trying to induce a voltage or a current into the antenna, into that cable. So they put a cable into the ground, drawing energy from the air. So they want to induce a small current into it. It would be forming a ground bond. You know how you have a bond or an earth for a house system, for instance? That's what that's forming. That's putting out a big bond. No power is needed. Yeah, that's right. But what it's doing is it's putting out a big bond. So it would be working with the cation exchange capacity and the voltages in redox in the soil, and it would be forming an equal pattern. So things didn't fluctuate too much out of that, because if that was bonded at around, this is pretty technical stuff, mate. If it had the EH is how you measure the millivolts in the cation exchange, and that would be forming basically a neutral or a buffer. So things wouldn't vary out of it too much and sway too much. So I reckon that's to do with the cation exchange and soil voltages. That's cool. That's how it works. The benefits out of it, the benefits would be that you're going to get this would be a really cool thing to discuss with when I was talking about the cation exchange and anion exchange capacity, because that's soil voltages, and it's all to do with what this process is. It's neutralizing it. It's forming a break barrier in the middle. That's about all I can explain about it. Hello, Dave. Nice to see you, Dave. Bamboo, no power is needed. Yeah, that's right, because they're just copper. Oh, hello, Deja Hu. Copper is a earth stake. Yes, that's right. It's just forming a big, massive bond area in your substrate. Wow, that's a good question. I like the technical stuff. Good on you, Bamboo. What's it called? Electroculture. They bury a copper ring and see what happens. That would be really good to keep I wonder, well, it's going to neutralize. So you could measure both ends of it and see what sort of voltages you get and see what sort of charges gets put into it. If it retained a positive or a negative charge, it would be around about if you put it into water, it might have a negative charge into it. So it might be reacting bad. But if you put it into dry soil, it might have more of a positive charge, and you might attract too many calcium or the cations, that you don't want to attract. That's a pretty interesting topic, that one. Oh, here's another question, something about it. Just plain copper wire, it's used for vegetable growing. Okay. That would be used to control the mineral nutrition or the elements that are in the soil, I reckon. It would, because everything's got a positive and negative charge. So that's what that's helping to do, to reduce that hardcore negative charge, which is going to bring in all of the elements that you don't want to go in without going into detail. How can I share this with a good screen? I wonder what thing I click. Oh, that one. Is it that one? Let's try. I've got my tablet up, so I'll present again. Share screen. I'll get back into it. Wow. was that one? Yes. There we go, it's full. Yay. Now you can see what I see. Attacked a plant, neighbouring a plant. Oh, this is how it speaks to another plant. So it puts out the salicylic acid, and then it turns into methyl, which is a volatile form, and it forms this way. The plant receives it through its receptor, uptakes it and transforms it back, and then acts its signalling pathways for that chemical that it's just received. Allelopathy. Oh, there it is. It turns it into a methyl step by step, changing to methyl when it's volatile. This is pretty cool to do with the hypertonic, isotonic, and hypotonic solutions, where one is very full than the other. It's called turgid when it's very full and flaccid when it's not full. These are cells. So it's to do with the solution that's inside of the cell. So when it's over full, it's hypo, it means it's adding to it, hypo, and then hyper is referring when it's leaching out of it and it's shrinking down. And the reason you want that is because that's how the plant reacts with touch sense. And this isn't really medical cannabis, but this is a mimosa tree, and that's how the plant reacts with that. So it loses the loss of potassium ions, result in hypertonic environment inside of the cells, and therefore it takes the water out and it makes it go flaccid, which makes it all limp. So that's why when you touch it, it reacts that way. Another way it happens too is through when you touch also the outside of cannabis leaves, they react with calcium, and there's a spike in the calcium concentration, and that amounts in calmodulin protein, which that protein then binds and then activates other proteins. Roots bend. Here you go. Here's a bit of vibration technology. Phonotropism. Phono. It's just showing here the sounds coming from the left to the right, and you can see that the root tip, the radical right down there at the root tip, the root apical meristem is moving towards the left, which is the direction the sound's coming from. This is a study done in 2012. Here's another study that was done in 2014 that shows the plant's response to leaf vibrations. So there was some herbivore was chewing on the leaves, and then the plants were responding to those signals that it was getting through the vibrations in the leaf. It was a very cool study, isn't it? Remember then the plants, they respond to a, what's that receptor? FLS1 receptor? FLS2, FLS, Remember, it's the flagella bacteria gene that's initiated in the bacteria that when on the plants can pick it up and it activates that FLS, I'm going to call it 1 receptor, to tell that plant that it's the type of bacteria has a flagella on it. So it's similar to this. So it's just activating different genes in the plant, because the cannabis plant has 35,000 odd genes. It's heaps of it. So here's another test done with no stimulus, and then that's the control up the top, and here's the one that had the 200 hertz frequency, bass and in, boom. That was done in 2017, that study. Phonotropism, growing towards sound. That's what that means. Another example of it. Calcium fluxes signal change to lead to the bend in the roots. That's how it actually gets the bend action through. I'm not going to go into that. No, very technical. Sound waves alter calcium fluxes. No, not going to talk about bending of the roots being there. Gravitropism. Gravitropism is like how it sounds, gravity. So entropism, which way it goes. So what's it go through? Is there a gravity response in plant roots? Yes, there is. So this is a plant root, and it shows the primary root here, and the secondary roots, lateral roots come off the side, epidermis, and the cortex is the inner layer, and the endodermis is the full-on middle layer, where the vascular tissue is inside of it. And carrot's also a root. Yep. And it's showing, what's this say? Observation. So it's saying that gravitropism, apparently through this experiments, happens through the root tips. So if you see here, the root tips under the root cap, and it's got to put out that root cap, because when a root goes into the soil or where its substrate is, it's trying to push through stuff, so it will get injured. And it doesn't want these cells in here near the quiescent center there to get injured, because then that will just destroy all its other growth and development pathways. So this experiment shows that they burnt it. So they burnt the tip. So here you can see these cells that are actually damaged, and then it went straight. So it's proving that the root cap there has the sensors in it for gravitropism. That was a rad experiment. Look at that. Oh, you're going, what is it? Hang on, we'll get to it up here. This is really, these slides are not done in order. I'm real sorry. It's a shame. The tip generates a soluble signal. Yeah. So auxin, this is talking about, it's another of the 5 basic plant hormones. It's talking about its action in the plants here for a bit. And Charles Darwin says, we must therefore conclude that when seedlings are freely exposed to a lateral light, some influence is transmitted from the upper to the lower part, causing the latter to bend. So he's saying, Charles Darwin in 1898 was saying that it's a polar, it's a what's it called? It's not polar. It travels from the root ship, from the SAM to the RAM, from the shoot apical meristem down to the roots. So it's generated up the top, and it's transmitted through the plant down the bottom. And remember, what was it last week, talking about the speed of it? It went 5 centimeters or 2 inches every. No, I'm not going to say that because I can't remember it. Soluble. Okay, next one, auxins, functions. Auxin has many functions. It's one of the primary hormones in plants. It's to do with the growth and development, branching, seed development, initiation, cell elongation. High auxin concentrations inhibit elongation, and lower auxin concentrations promote elongation. So to do with when you're doing tissue culture, this is why you have to get the right amounts of the tissue culture. You use auxin, and then you use cytokinins. So this is to do with the roots, and cytokinins is to do with the shoots. And if you don't get the right amount, this is what can happen. It stops the actual works against it. So you think you're putting heaps of auxin in, I'll put more rooting powder in. It'll work really good. It doesn't. It actually stops the growth, reduces it. And what's its effect? What's auxin's effect when it's in the sun? So it elongates. And how does it elongate? It's because it moves to one side in the cells, and it makes these cells go longer. It's stretching the cells out. So much it stretches those ones out there, it forms they're longer than the shorter ones here, making it trope over. Auxin has a polar response. That's what I was looking for before. I was muddled. Sorry. Auxin is produced. Yeah, that's all right. Auxin moves to the roots independent of gravity. Yeah, so see, if you grow horizontal, it'll still work fine. You can still maintain auxin maxima. Auxin maxima is the process that you want to maintain the maximum amount of auxin production. Auxin response coding. Oh, this is getting into a bit technical stuff. Oh, that's the blue. This was stained. Methyl was stained with methyl, and you can see the blue response there, showing the auxin accumulation in the meristem cells. The conclusion: auxin is produced at the root tip. Oh, geez. Auxin. Okay. Sorry about that. Sensing gravity. So wild type and gravity mutant. So mutant, that would have no gravity response, so it will grow outwards. And the wild type has a gravity response, so it's going to come out and has a gravity effect with it. Concluding that some of the mechanisms of gravitropism may be different in the stems than they are in the roots. Angioplasty, no. Observation, second, no. Doesn't happen in plant. In cannabis, plants have an ability to retain past events. Really? It's our, what's it, our hippocampus in our brain. Do plants have hippocampuses? No, but this experiment, I can explain that one. There's a better one. There's one where they stabbed the leaf. Morphogenic memory. I type, oh look, it's a different thing. How do I understand this one then? So the inhibited lateral buds are there, okay, developing down the bottom. Now, lateral buds released from inhibition, they're all released, and now what's the memory say? And then the memory shows that they were still inhibited before, and it doesn't release them. In the morphogenic memory, it's a type of memory that later influences the shape or the form of the plant. So this one grew, that's the control at the top, how it should grow. Down the bottom, it grew like this. It grew from that, the next one was that and that. Why? Because they put a stab through the center of it, and then what it did was, and they took it out, and the next time through its breeding, the plant remembered which leaf had been damaged, and then it only grew on the opposite leaf coming out of that because the left one was damaged, proving that plants do have long-term memory. So be careful with your plants. Don't thrash them around and all that sort of stuff. You know, they do remember, they do know what's going on. I wonder if there's any questions. Actually, I can see my tablet. Momanic Meds, how you going? Nice to see you. Vernalization. It's a transition through winter, allowing plants to flower. Vernalization is another way of activating their flowering pathways. You know, there's a few ways to doing that. Through photoperiod mainly, you activate it and the FL1 gene turns on and the floration starts to happen. This is another way that you can do it through vernalization, which transitions, it's to do with temperature. The FLC gene is active and inhibits flowering. Oh, this is going to get into it. The transition through epigenetics. So as epigenetics happens, generation 1, then it changes again and it changes again. There's a good, if you're interested in cloning mother plants, look up the genetic mosaicism hypothesis, and that's a rad paper that they go on about where to clone at the top, middle, or bottom. Where should you take off to retain the maximum genetics of the plant? Because epigenetics is a massive influence. It happens everywhere. That's how things evolve. And if you don't want your plant's genes to evolve by the conditions that they're kept in, you should take them right in the middle to the lower bit. That's where you take your cuts from. So for your next mother plant, that's where you'll take it. But if you want one to vary heaps, go from right to the top, and it'll be like 75% different. So you get a different phenotype will come out from your mother plant through epigenetics. Epigenetics hereditary. This is to do with breeding. Next week's going to be on breeding. I'm going to get right into it. I'm going to open back to the cool things where I used to show all the latest news and that sort of stuff. So next week, I'm going to talk about breeding. All those slides that I had, bang, it's going to be next week. So I'll get into the hereditary genes and how genes can turn off in different circumstances and turn on in other circumstances, and what that result is, and how things can like skip a generation and that sort of stuff. So back to this, what does this show? This is control, so normal after 2 weeks, salt stress plant. Okay, so by putting in a normal plant, you get this after 2 weeks. If you salt stress it, so you put this mixture and you put it in, and 2 weeks later, you'd expect to get that normally. That's what that's explaining. This is a bit, what's this explaining? This is showing that control, they did the same. So they put a control plant in a high salt mixture, nothing. They went and get a salt stressed plant and they'll put it in the mixture and they start to see growth in it. These are little calluses. So they're doing some marker assisted selection to see which offspring they'll get will be salt tolerant. So if I was to get those genes from Jono, for instance, and breed with those, I would do a pheno experiment where I would put out my first generation, so my F1s, or it depends if I crossed or did an S1, which is an inbreeding cross, and then I would put my first generation, I'd do this experiment. So I'd put in a high salt mixture, and this is how I'd pull it out, and I'd find those genes that were still growing by doing this exact thing that you've got to do here. If you want to find different resistances to different pesticides, different things like that, different drought, I've done fungal, I've done quite a few different things. You've got to put them under those conditions and see which ones survive, and then you know, bang, those ones, and then test it again and make sure epigenetics doesn't swing in too much and turn those genes off. But this is a way that's done. I won't go on about that. Aussie, hey, nice one, Stony Creek. How's it going, mate? Breeding, look forward to it. Yes, it should be good. It's a bit away of marker selection as well. Memory and what? Consciousness. Oh, yeah. Memory occurring to endeavor tolving. I don't know what that means. Procedure, not memory and consciousness. So forming of the memory by stroking the plant, retaining the memory 30 minutes later, and then recalling the memory by something. Okay, I'm sorry, I can't make that out. The lowest level of consciousness characteristic for predominant memory, too many big words, level of organization. That's the growth and development. That's not related. They're different types of rad trichomes because we're trichome farmers. You don't want these unis. You want the big bulbous. Oh, geez. What is this in here? Anyway, it's cool, isn't it? So showing different types of cells that are inside of a trichome. I'm going to name them. Very cool. So you want to try and fill this up and get a big light microscopy image of a trichome following thin section sustaining. Okay. This is technical stuff. Sorry, plastids. Okay, good. The number of chloroplasts vary in different cells. They sure do. Chromoplast, the red pigment. Pigment plastid. Okay. What are we sort of getting to here? Yeah, there's different types of the starch ones, the amyloplasts. That's why, remember, you can mimic that by adding H2, iron pyrite for germination in the seed process, because starch is one of the biggest things to be broken down in the seeds. So to help the plant, help the seed do it, you can add the H2O2 also does a little bit of it, mimicking, and the iron pyrite. Chloroplast movement maximizing, yes, that'd be cool. Hey, that'd be cool. I've got a rad compound microscope. I can show you this. Not now because it's not set up. It takes a bit to set up. But I'll pull out some cells. I'll do a microscopy one soon. And we'll pull out some plant cells. And what happens is they dance around quite fast, not like the northern lights that go slow, but they dance around. You know, you can see them moving anyway. They don't dance. And then they'll go they'll move to one side, they'll change to sides and stuff like that. And that's what this is talking about. The chloroplasts, they move to maximize their absorption of the light. So see, there's heaps of light there, and they're spread out in medium light and intense light. There's not many of them at all. So by thinking you're putting in the plant with maximum amount of light, you're going to get maximum benefits. It's not. The plant is limited by its lowest nutrient or factor. And that's by Liebig's law of the minimum, it's called. And so with the photosynthesis, this is regarding. So photosynthesis, there's 3 things to limit it. And there are temperature is one of them. So you want to keep it at about room temperature or a bit smidgen higher, depending cultivars. And CO2 is the other one, because remember a plant's 17 essential elements, 3 of them come from the air, making up 95% of what the plant requires. And that's one of them is carbon, and hydrogen and oxygen to the other 2. And 45% comes from carbon. So for it to get its CO2 is a good way, most of the way where the carbon gets into the plant. So if that's one of its limiting factors, it's can only do as good as the light. So it's light, CO2, and temperature, the 3 limiting factors for photosynthesis. Yeah. Mitochondria and chloroplasts. Oh gosh, it's getting good. Yeah, because they've got chloroplasts in the mitochondria. The origin of chloroplasts. Okay, no. I want to see. Yeah, it's called the cell walls. Vacuoles, no, I'm not getting into that. Plant cell elongation, touched on that before. Trying to keep it interesting. Vacuole plasmatospires, no. The cell membranes. Oh, that's how cells speak to each other, I suppose, through the plasmodesmata. That's a little gateway that links them up. That's what that's sort of talking about. Tissues. Nope. Do you want to know about the tissue? Leaf, stem, and root tissues. They're all pretty similar. They got outside, inside, and in the parts that they move and breathe with. So they're the parts like where the vascular bundle is, the xylem, the phloem. Here's something interesting. Epidermis is the outside layer. That's what it looks like under an electron microscope. You've got these little bumps. They're the cells. The trichomes growing out. Trichomes have many functions. Xylem. Sorry about this. Cell, yeah, our cells are divided. This is talking about, I want to not get technical and keep it interesting. This sort of gets a smidgen technical. This is a cross-sectional layer of the stem. Looks pretty cool, eh? You can see that's a dicot, not a monocot. That's the different types of, anyway, you probably not. That's how the water and the elements get through the plant. Water is the xylem and the phloem is around here. Okay. Cannabis is an annual plant. Water and mineral uptake by the roots. Very good. Sorry. Shoot development. No. Pretty fascinating how it happens though with shoot development. Flower development. Yes. It's the flower. Chloroplast, how it photosynthesizes. They're I talked about it before. It's the energy that it makes in the chloroplast. That's how it functions. Cell's energy currency is the ATP, adenine triphosphate. That's another NADP. What's his photosynthesis? First reaction. The chemicals exchanging through light reactions and through dark reactions. Oh yeah, it just goes through different cycles. There's a dark cycle and light cycle because this photosynthesis also happens in the dark, believe it or not. Oh, yeah, it's where it has stored energy. When chlorophyll absorbs light, the energy from the light causes it as an electron to jump out of the chlorophyll, and the electron travels in pairs. Wow. So it's, okay, that's inside of the dermal tissue under the leaf. That's dermal tissue. See the cells on the outside? It's showing their polar orientation. Okay, it's not really explained much. Oh, here it is, where the energy is traveling from one cell to the other. And that one's full. The chlorophyll in photosystem 2 donates an electron that's transferred to photosystem 1. That's a cell wall. It's a cell wall inside of a cell. And photosystem 2 and 1. That's, well, it's mostly the photosystem. That's really technical stuff. Here's a question for you. What is the source of oxygen in the air? So how does a plant get oxygen? Does it come from the soil, or is it through the stoma, through multiple stomata, or is it CO2 from the air? So how does it get it? It gets it from the soil through the breakdown of the molecule, the water, and it gets it from that. That's another way, actually. There's two ways that you can get it. You can get it through the roots by absorbing it, and another way you can get it through photosynthesis, and that's where it splits the hydrogen with the 2 oxygen and uses it that way. Most of it comes from the out of the roots, though. That's a bit technical. Looking at the chat, Jimmy Growtron. What's up? How's it going, Jimmy Growtron? If you're interested in growing, Jimmy, next week we'll be talking about, actually I'll show it. Yeah, I'll stop this for a sec. Aussie, it's happening, breeding. Woo! Yes, look forward to it. Yes, I'll show you actually. I'll just find it first. Next week's. Because it's cool. Because I've been just saving it for so blooming long, and finally I thought, right, it's on. I'm in more of a organised frame of mind now to get more organised, I suppose, so I can get back into all of the fun lives where I was having all the research, all of the country developments, all of the latest things that happen, that type of stuff. Where is it? Plant genetics. Here it is. Check this out. This is what I plan to go through next week. I'll probably show this a few more times because I love this. I put this on hold because I really wanted to. That's not it. Genetics. All of this. Look at all that. Wow. All these rad things that talk about genetics, different things that happen through breeding, how to test the breeds, how to test your different crosses that you've done, how to pheno hunt those different crosses. You don't need to put out 100 seeds, one bit. You can put out 3 or 4 or 5 seeds, such a small amount for a pheno hunt. How to do all these that's through marker-assisted selection, if you use those types of things. I'm just going to touch right into it through the testing and all the HY5 gene also, which is the elongated hypercotyl. Let's go into that a little bit because you really want to know about those ones. Or you want a DWF gene, which is the opposite, which I've had. How do you know if that's dominant in genetics? Well, you just do these tests where you put 3 out, and if 2 of them come back with the trait that you want, that means it has a dominant gene. If only 1 of them come back with the trait, that means it's a recessive gene. So I'm going to go into all that stuff next week and get into it. Again, it's real fun. I love breeding. It's the go. Woohoo. So it's next week. Hey, martial artist, what's happening, fella? Jimmy said, how you going, Jimmy? Yep, didn't miss you. Bamboo, Flora Plus Shuffle it was. I'll see you, martial artist. Well, I'm going to get back into this little bit here. Today, it was an open topic. There wasn't really much suggested. So I started talking about what a plant knows and how it can hear and touch and feel and that type of stuff. So I'll get back into that and finish off. This one, the ATP. It's been through adenine diphosphate changes after it goes through its reaction. You know, that's pretty technical. You don't want to know about that, eh? Inside all that. Let's talk about plants were domesticated in several regions. I don't know. Human intervention has invented the colonized. Yes. Yeah, did you know that this is a bit off cannabis, but from wild mustard, this is a show about genetically modified plants, GM plants, on how nearly everything is. It first came out as wild mustard, and then they fiddled with it, and it made cauliflower, broccoli, cabbage, kale, and kohlrabi, an onion-looking thing. So it made those 1, 2, 3, 5 vegetables out of the one mustard, excuse me. Central dogma node. This is DNA stuff. If you want to know about the DNA, there are 4 types. GATC, the names of them. I'm not going to name them. Proteins are made. Yeah, good. And again, cytokine for growth omega. Okay, what's that? Recombinant DNA. Oh, that's how to grow DNA. Gene transfers, genetic engineering. How can this be good? Oh, that's it. Okay, very good. Oh, I see Dave's written something down here. What's Dave got? Hey, Aussie, have you noticed the seeds with all these new genetics in the last 15 years are getting smaller, or is it just me? We can talk about that for a bit. How long has it been going for? There's no timer thing here. There used to be a timer. I don't know how. Oh, well. There it is, 47 minutes. Okay. Yeah, I'll show you something about the seeds. I did an experiment with some seeds. Also, Gregor Mendel has done some good seed experiments, and their seed size, it's proven that it doesn't, he proved it, that it didn't mean much on the outcome of the genetics inside of it. And I've proved as well. I'll have to find this first. Actually, I know where it is. It's under seeds. Seeds. Yes. What was I going to say about the seeds? Oh, the size of them. That's right. I've got it. Where's that chart? You're kidding. It's not here. I've done some really good. I've got the chart. All right, I'll show you this chart. It's not the right one, though. These, I haven't noticed, I don't buy a lot of seeds, I'm sorry. But the seed size doesn't really have much effect on it. Oh, Dave says, we used to get seeds that were as big as apple seeds. Oh, yeah. The reason being, mate, is because the seed size, they might have gotten smaller now. When people have kept those genetics, the seed sizes that have selected has been smaller when they've put the progeny out. What am I showing you here? This is, it's a normal seed size. What's it? 1, 2, 3, 4, 5, 6 centimeters. 6 centimeters? No, that's massive seeds. These must be the ploidy seeds that I made. These are tetraploids. These have extra sets of chromosomes in them, and they're abnormally big. As you can see, that's 1/4 of an inch for those people in the States. There you go, a bit over 1/4 of an inch. Oh, good. And there's the other ones up the top. And these are some 1-to-1 seeds I made. So I crossed some, a keeper of CBD. It was a high CBD strain. It was like 20, low 20s it tested. Really, it was really 20%. It was just so high in CBD. So I crossed that one, and I kept the, it tasted like orange sherbet. So I did a few crosses to make some 1-to-1s, and this is a death bubba I crossed it with, and that's the difference. So you can see what, 1, 2, just over 3 mil, and this one is 1, 2, 3, 4, 5, 6.5. So just over double the size of it. That was a pretty cool seed experiment. And the offspring out of these were perfect. The genetics are fine in them. If I was to do a DNA test, I'd find that all of the, at least probably about 35,000 genes would have been retained through both. Oh, there's the conclusion. 2.5 mil up the top and 6.5 down the bottom. It's a massive difference. It's pretty cool, mate. So yeah, and it didn't make any difference out of the seed. It was just to do for the seed size. But what it does, I tell you what it does help is the seeds that are this big, it helps with retaining the genetics to making sure that they're going to have an outcome, because there's more starch in here, so there's more of a chance that this down here in the embryo section, it will do its process that it should to have it germinate. So there's probably something in here instead of me just. It's not really what I wanted. Kind of that. Anyway, that'll do. Stony Creek says, I heard original landrace seeds have bigger seeds. Yeah, probably because of this, they just keep breeding at the same size. They have struggles to get out and reproduce, where these ones here, that was an indoor grown cultivar, and its chances of failing are low, where it didn't have any need to make it massive. Yeah, it was just a trait. Also, I've got another one, Island Pink. It's a trait of small seeds too. So if I was to cross with that, which I have, I've gone and pulled the bigger, like pink death bubba. So I would have crossed a smaller Island Pink with a big pink death bubba, because they were big, the death bubbas. And then I selected ones that were large out of that stock. So then the offspring out of that would be large. That's how I've got them. But say in this one, I could have got one of the small, see this one, bred with this one, sprouted this one, bred with that, and pretty much 50% of the genes would be influenced to have small ones this way. So you could do a marker selection out of that, marker-assisted selection, and get what result that you wanted. So if you're a hemp farmer, for instance, you'd be only breeding this way. But if you're a farmer where you needed space in your lab, and you might want to go on the smaller side. Jeez, that was pretty cool. Seed stuff's good. That's breeding. Next week. Ooh. Oh, what else we got here? Oh, yeah, heard that's right. They used to be that one. And the larger seeds at landrace. Yep, because the landrace, they would have struggled to try and regerminate. It wouldn't have easy pickings. So it would have had to go bigger and try and reproduce more of itself to have a successful outcome. Yeah, very good. All right, 54 minutes. What else can I talk about? I'm here for you people. Please make a suggestion. That was good about the seeds. Thank you for that question, whoever said that. I did a course called Seed Science and Technology, something along those lines. No, it's not science. It was seed farming and technology. And it was quite interesting at the start of it. And then at the last bit of it, it got into the, I suppose I can talk about seeds for the last little bit. All right, let me pull up my seeds. I'll pull up the seed course, see what it was called. Principles of Seed Technology. There it is. Do you want me to run through this a bit? You want to see about some seed stuff? Okay, I heard everyone say yes. Okay, no worries. Here we go. Let's run through, just see if it's interesting. Well, it was for the first few weeks. Uh-oh. That's not it. Week 1. Okay, it's a seed and what's inside of it. There's varying types of seeds. This will probably go into a bit of the history a bit. The seed coats on the outside, or the tetsa. Dormancy. Yeah, all seeds start out into their dormant stage. These are pretty cool. These are synthetic seeds. I got a video I've made on how to make them. You use calcium carbonate with not even going to say the next one. You use those two and then you mix them together, and once you drop one into the other, it forms this little caviar, little oyster, little ball thing, and you put your tissue culture into, you sterilize it first and then put it into one of the soaks and then put it into your little dropper, and once it drops through, you end up with these little balls, and they're synthetic seeds. So they will go out, they've got everything inside of them to the right vitamins, nutrients for them to successfully germinate. That's cool as. Microbeads, I think it's the video that I made on them. That's how fertilization happens. So I'll talk about this breeding next week, which is pretty snazzy. How the pollen sac, it comes down here and has calcium signals, goes all the way down here through the micropyle on the end of the ovary and fertilizes. And then if it goes a little bit further still, if there's a second or a third sperm cell, it can go through and pollinate other cells inside of the ovule here, and the ovary, sorry. And in the center ones, they've got 2 chromosomes. The end cells have 1 chromosome, this middle one has 2, meaning if it gets into that and pollinates that, that's how one way Mother Nature produces triploids. There we go, double fertilization, just what I explained, but in a chart form. It's only I was on this page. Anyway, see how the pollen grain lands in the stigma, travels down the style, and if it's got a few more sperm nuclei, it can travel past the central cell, or the mother cell, it's also called, fertilize it, bang, got rad triploids. Triploids are the way they're about 90% infertile in nature. And if you breed with a triploid, you will get a tetraploid. And that's what and now I'm not talking about breeding. I'm already talking about breeding. I love it. This is the embryo sac. So that's how the sperm cell comes down through here, through the micropyle, and comes in here and fertilizes it. Parthenocarpy, no. Testar, that's the outside of the cell. Actually, that would be good to talk about. Not the pericarp, no, we're dicots. That's how it forms inside of it. That's the inside of the cannabis seed. That's what it has, a lot of starchy stuff. Where's the pathways to activate it? Seed maturation. That's another way to check your seeds, to see how they mature, your rate, to make sure that they come out correctly. And there's a few things to be a breeder, successful breeder. There's a few criteria that you want to tick. Let's do, yes. Okay. Actually, that's a rad chart. Where's the cannabis one that I made for that? This one. So, this is with breeding again. I should talk about this just next week. This is an example of what I'm going to talk about next week. So if you're, when you're breeding and you're looking for your development of the seed, how long do I do it for? How long do I make it for? If you want to induce it, how long does it take for it to produce the pollen? How long does it take for it to produce the seed if I go and manually pollinate it? And so what happens if you do it then? In 10 days' time, it'll start and do its thing, and it'll be ready by about day 25. So whenever you start it, you'll be getting along, and you might see some other, actually I've got photos of those green seeds. Anyway, I'll talk about more about that one next week. I'll get back to, what was I going to share again? Week 1, right? Going through a little bit of the seed stuff. So yeah, I'll talk about things like that next week, because you want them to develop. Yeah, otherwise they won't develop properly, and you'll get your immature, you'll get your green ones. Actually, there's a few questions there. Not questions. There's a question right there. I better go back. Hang on, I'm just going to pause this. Martial artist has got something. I heard landrace, yep. Martial artist has got seeds from a friend farm in Morocco, and they were the tiniest ever seen, and they're still the most vigorous. Yep, that's good, mate. Seed size, this martial artist says, seed size seems to have a lot to do with the size of the bracket, which makes a lot of sense. Okay, that's good, mate. Stony Creek. Did you make hash out of the Moroccan? Okay. Oh, that's probably just someone else, because that's I didn't have any Moroccan. We're talking about the seed stuff. Oh, that's right, that seed. How long got talking? Been talking for an hour. That's long enough, eh? Or do you want me to talk a bit more about seed stuff? I could nearly do a whole, because the class, it was an 8-week course, so I could do no worries an hour just on seeds. So maybe I should really save that for another show. Seeds, dormancy, all that sort of stuff. I've got into a few of my videos to make some of those on the dormancy, because there's a few rad techniques you can do to trigger those pathways, the gibberellic acid and the ethylene. That's in the pathways for the seed. Where's that one then? I'll find a pathway just to show you. There's one. Even though this probably should be talked about next week. I've said that a few times, but I'm still going to show you, because it's cool and it might help somebody. So this shows the gibberellic acid and its signaling pathways, so how it helps with germination, and how abscisic acid, which is another one of the basic hormones, it works against it. So the more abscisic acid, the more dormancy it keeps. So this is a dormancy breaking slide on how to, so it's gibberellic acid and ethylene are the two plant hormones that are involved in the germination of a seed. So if you can add them to your solution in the amount that it needs, in a smallish amount, you can help it germinate. It's another technique of breaking dormancy. Pretty cool to manipulate plants' genes like that, because plant has inside of it, its cells, it had an embryo to produce that seed. So it has its mother's cells inside of it. And I did a tissue culture experiment a few years ago where I made the seed produce a root out of the mother's cells, and then the seed still germinated properly. That had the pathway that it was still initiated for. So then I divided them both, and then I ended up producing the mother and the daughter out of one seed, just by reproducing, multiplying cells, by using hormones, more so hormones. So it's pretty. Oh, Bamboo, thank you for your support, mate. Good on you. Appreciate that. For anyone else who wants to support, there's a subscription file there. There's 4 different categories. One's as a thank you one for the top one. Second one's for the playlists that I haven't put on my playlist and released them. Under my playlist, I've got thousands of lectures related to plant microbes and soil science for free for people to look at. And the third tier is for $100, and it's for someone who's starting out, and then 100 who just wants minor help, and then the $300 one is for somebody who wants full-on help. There's a few things in there. So that's the subscription, and the little thank you buttons down below, the little super chat or thanks button. But appreciate it to all the people that helps. It does help me a lot. I put a bit into making these things. Even more next week. Look out for the breeding one, Bamboo. It will be good. And you'll just be seeing me getting excited, and it'll probably go way over like normal. What's Stony Creek say? Oh yes, AussyCC. One of them would be a clone of the mother plant, right? That's interesting. Yes, it's this. Actually, probably the quickest way to explain it is to nearly just show it, because I'll see if I can find a quick snippet of it. Your channel, because it's the embryo cells you want to reproduce. What is it? That one was done years ago. That'd be right at the start. Yeah, here it is. How do I show this? Oh no, just press play and then share it. So I'll share my screen now. Oh, this takes off views from me. Anyway, this is the, good on you, Bamboo. Thanks, mate. This is I did a world's first in tissue culture a few years ago. And it's this one here. And you can't hear it. Why not?
Her seed and still produced her daughter by organogenesis. And that organogenesis is the way you use tissue or cells to produce roots or shoots under different types of side there in cotyledon leaves still in the center. And then the radical triple response, how it's got the hook and the thickening. And then you can see it's still going to go and sprout, no worries. So I just had to take it off, separate it, and then I had to change the medium and then put some more cytokinins in to produce the plantlet, and then it went fine.
Because remember, you want the cytokinins and auxin. They're the only 2 hormones you really require in tissue culture to get it cranking.
And then this is how 5 requirements for a plant cell: inorganic nutrients, carbon source, vitamins, growth regulator, and organic supplements. And that's what you need for plant cells. But you have to get the right amount of hormones right, because high amounts of auxin produce roots, and high amounts of cytokines produce shoots. So here's some formulas for you to play around with. So you can see in there, that's auxin up the top, kinesins and cytokines. So there's 2 things, but sterilisation is the key. So if you don't get sterile, holy mackerel,
you dip them, for the sterilisation, dip them in the isopropylene. Use that a lot inside of your laminated flow hood, or if you haven't got a laminated LFH, you just cut a few holes inside of a big box, tip it upside down, and 10% bleach in there.
Holy mackerel, you can go and grow fungi and bacteria. Like have a look here down the bottom, see the cultivation time, 2 to 10 days for microbe cells, 2 to 4 weeks for plant cells.
Massive difference. So if it's not sterile, you're going to massively double and triple your microbial population, where you only want to brew doubling your plants.
So it's hours to grow microbe cells and days for plant cells. So if you don't get the sterile right, and then once it produces organs, you're going to start and change the medium, and then you get
the chunky grey thing. That's the orange thing. That's the callus. It doesn't know. They're parenchyma cells, they're called, and they're undifferentiated, meaning they don't know what they're doing. So you've got to give them some direction. So you can put some...
And start to get different shoots and roots come out to the plant.
You'll add oxen for roots, if you want roots, or shoots, if small cytokines like this down here. And if you want both, to have a good medium blend, you'll add a medium blend of both.
And this is cool synthetic seeds, microbeads that I made with some tree culture. So I was waiting for them to produce some organs inside, and then I'll go from there. I made an oxen mix. So I would just produce roots first, and then I can go from there. That's another video. Yeah, I'll post that link. Pollen culture from microspores. So this was pretty cool. This is how I did it. So anthers produced pollen grains, and I cultivated that, changed the medium, produced that, and it has one set of chromosomes. So I had to induce it to produce two sets. And the method that I did was the float culture method, because I don't have a centrifuge at the moment. I got no lab. If you use the cell anther walls, you're going to have two sets of chromosomes where pollen grains have one set. So you have to keep that in mind, because I use pollen grains, so I didn't induce it. And this is cool. I just cultured just a leaf, and I like the fruit coming out of it under the microscope, so I thought I'd share it. All right, thanks for watching. Enjoy.
There you go. Yeah, good on you, Bamboo. Oh, yes, that's right. All right, Bamboo, no worries, mate. Oh, good on you. Thank you.
This is some cannabis.
Thank you. That's good. I try hard. Yes, the Australian community hasn't been too favourable. They've been spreading a few bad rumours. So if you hear some stories, they're stories, my friend. So, yeah, a few rotten eggs in the Australian community. What's martial artist? Had to run to the store. I'm 12 minutes behind, but don't want to skip ahead to see the seed topic. I'll catch up somehow. Yeah, the seed topic. Well, maybe I should have a thing for you. Maybe I should. Try and remind me, martial artist. I have some seed stuff down the track. But next week, I am very proud to say that I'm going to have the breeding. It's going to be all about the technical breeding, how to check your seed stock to make sure it's solid. So many breeders these days, they don't include resistance, seed resistance. You need resistance for things. Did you know that they'll even breed them in conditions that are in sterile warehouses where they don't use any sorts of biologicals, where they just want to remain all sterile, use hair nets, no microbes can get in. That means that the seed stock for that, there's a high chance that it's going to fail. And not a high chance, but there's a comparing that to organic seeds, because organic seeds have all the endophytes inside of them. So when they come out, they've got the microbes to already help with the different pathways and initiations, and they'll block all the other bad pathogens and problematic microbes that want to come in. So that's what I mean by the failing. They'll easily get through and germinate, but the maturity rate or the amount of time, the amount of success you'll have, or even the success you'll have in bad conditions, or that sort of stuff is what I was getting to. Sorry, I won't go on. I love breeding. Good stuff. Thanks for the support, Stony Creek. What's Dave say? These rotten eggs are keeping their heads down. Don't worry about those idiots. Oh, thanks, mate. Yeah, I don't like the drama. What a waste of time drama is. Look how much fun we've got. It's so much cool to talk about. Yeah, stuff the drama. Yes, I have followed a bit of gossip online, but it's easy to weed out the ego. Exactly right, mate. While I'm on that topic, I emailed a university where that professor says he's at, and they haven't heard of him. It's such a shame. I don't know why people lie. I don't want to get into it. But yeah, good cannabis science is what we need. Stuff that's why I just try and shoulder slides, so then it can relate to different things too. It's not just me crapping on about stuff that I might have forgotten about, or, things years ago. So it's all right. Next week, there's the thing I was looking at. I'm going to show it. This is what I was looking for the whole time, the easy germination process for a seed. I was just going back through my seed stuff then. This is just showing how gibberellic acid promotes seed germination. So with your ethylene, like before, with your out of your fruit, and the apples and bananas are the ones that secrete ethylene the highest out of all of the fruits, pears as well. But you'll put some fruit in with the seeds, but you can also go a smidgen of gibberellic acid, a very minute amount. Like if you're going to use a research level, you want granules. So you count out like maybe 9 or maybe 4 granules into a little tiny solution and put a few seeds into that to help. So the breakdown of the starch in the endosperm is initiated by gibberellic acid produced by the embryo or added during the molting process. So that's where if the plant could be recessive in gibberellic acid or have a mutant problem and it won't produce it, so that's another way that you can get it out of its dormancy. And the amylase is the enzyme that produces to break down the starch, because remember the amyloplastids, they have the starch inside of them, so you've got to release the amylase, and that breaks it down, the starch, which produces the energy and gets the energy happening. And the way that you can emulate this amylase is the H2O2, a smidgen of that, or of the iron pyrite is another, because the H2O2 and iron pyrite, it reacts with the water and then it forms this amylase molecule. And the aluminum layers, the outside. Yeah, but anyway, I just wanted to see how basic this is. So this is the endosperm, embryo. This is from the parent cells. And then all of this is from the cells that were produced from the donor, because you can also have female induction. It's not just a dioecious plant. You can have monoecious too, where it produces both the male and female. So yeah, just those different. I just like it because it's very simple. All right. Thanks. Oh, there's a bit of chat here. Can't wait till next week. Very good. Spot on, Bamboo. Yes, I agree, Bamboo. Spot on with what you said. Can't wait for next week. Yes. Martial artist. Oh, yep, they're talking internally. Very good. Oh, something about breeding. I'll read it. I'm not going to go a few season, maybe a few planning as breeding too. Yeah. Oh, yep. We'll save those questions for next week, mate. Thanks, Dave. Good one, Marshall. Sounds good. It definitely does. Next week, we can have all your breeding stuff. I'm excited nearly already. All right. Well, I appreciate everybody for rocking up and asking those questions. It was fun today. And especially thanks to Bamboo again for your subscription, mate. That goes a long way. Good on you. And to the other people who subscribed, I really appreciate that as well. Thank you again. And next week will be breeding. Woohoo! Difficult cannabis breeding, the technical stuff, and how to select it, and how to pheno hunt properly, and how to select things properly. Good stuff. Thanks again. See you in 167 hours. Happy breeding, happy growing, and good health to you all. Bye-bye. Thanks, mate.